So this is chapter 17, in which we're going to talk about the special senses. This is another one of my favorite chapters just because I love how the eye and the ear work. So we're going to start with a brief introduction and remind you of some information we learned in chapter 15. So recall from chapter 15 that the special senses, which is the focus of this chapter, differ from general senses in that they include olfaction, which is smell, gustation, which is taste, vision, equilibrium, and hearing. They also have special sensory organs localized to a specific area of the body. So, for example, vision is only carried out by your eyes. So a good bit of this chapter is going to be focused on the structure of those special sensory organs. They also have more complex receptors, and again, a good bit of focus in this chapter is going to be on the structure of the different types of receptors. They also have their own specific cortical area for processing. So with the general senses, all of the information went to the primary sensory cortex and the parietal lobe, but each of these special senses have their own cortical area. For example, visual information goes to the primary visual cortex and the occipital lobe. Now, some things that they do have in common that we already covered in chapter 15. The stimulus must be transduced into a potential. So that means the stimulus has to be turned into an electrical signal that neurons can carry. And so again, a good part of this chapter is going to be focused on how these complex special sense receptors actually do the transducing. The receptors are also very specific, so a visual receptor will only be stimulated by light. And these receptors also have receptive fields, so that principle still applies. They also have specific pathways that go from the receptor to the cortical area, and this is how the brain interprets the modality of the stimulus. So this is what we call labeled lines. That is also present with your special senses as well. And adaptation is possible both at the peripheral level and the central level, although it can depend on each specific sense, and we'll talk about adaptation where applicable in each specific section. So in this section, we're going to go over olfaction, which is your sense of smell. So you have two olfactory organs on the roof of your nasal cavity, so one on each side, one in your left nasal cavity and one in your right nasal cavity. And this picture up here shows you the olfactory organ. So it's the part that's actually embedded in the roof of the nasal cavity, and then the structure above it is called the olfactory bulb, which you looked at in the sheep brain last semester, and we'll talk about the bulb when we talk about the pathway. So the olfactory organ itself has two layers. The first layer is an olfactory epithelium. This layer contains the olfactory sensory neurons, which are shown in this image as the light blue cells. It also contains supporting cells, which are shown in this image as these pinkish colored cells. And then there are also these basal epithelial cells, which are stem cells that can divide to replace worn out olfactory sensory neurons. The other layer of the olfactory organ is the lamina propria. This is that connective tissue layer that sits underneath the epithelium. And the lamina propria is notable because it contains these olfactory glands, or at least it contains the base of them. And these glands are actually exuding or secreting substances which can then absorb water, which then forms a thick pigmented mucus. And this is the mucus that you find in your nasal cavity. So why do we need mucus to smell? Well, first, the mucus helps keep the epithelium from drying out. So this is a delicate epithelium. It's not hard and keratinized like the epithelium of your skin. And there are also proteins in the mucus that can bind to hydrophobic odorant molecules that allow them to dissolve in the aqueous solution so that they can bind to the little receptors on the ends of the olfactory neurons. And there are proteins in the mucus that can break down these little odorant molecules so that you have room to smell new ones, right? So if you imagine all of these little odorant chemicals getting stuck in the mucus and staying there forever, you would never be able to smell new things. So there are enzymes in the mucus that break these molecules down, which make room for new substances to smell. So let's take a closer look at the olfactory receptor itself. And again, in this picture, these are these long, skinny blue neurons. So the olfactory receptors are themselves sensory neurons. So they are highly modified sensory neurons. And there is an extensive turnover. So as they wear out, and the reason they wear out is because they do have these extensions down here in the mucus layer. So they undergo a lot of wear and tear, so they can wear out very easily. And then you have the basal epithelial cells, which can divide to replace the worn out sensory neurons. But do note that even though the basal epithelial cells are replacing worn out sensory neurons, as we age, we still have a total reduction in the number of these olfactory neurons, and they become less sensitive. So your sense of smell does decline as you age. It is also a symptom of some diseases, like we know that in Parkinson's disease, your sense of smell is one of the first things to go before you ever notice any movement problems. So if we go back to the structure of the olfactory receptor, at the very ends of these modified neurons, there is a dendritic bulb that actually protrudes out into the nasal cavity, and then these bulbs have little cilia that extend away from them and go into the mucus that lines the nasal cavity. And the purpose of having all of these cilia is that it provides an extensive surface area for the chemoreceptors that are actually embedded in the plasma membrane of all of these little cilia. And so here's another image just to give you an idea. So in this image, we can see our olfactory neurons are all pretty colors. They're green and blue and red. And then if we take a closer look at the little cilia, this is where we are actually going to find the receptors in the plasma membrane that the odorant molecules are going to bind to, which we're going to take a closer look at on the next page. All right, so let's see how the process of olfaction actually works. So it all starts with the dissolving of an odorant molecule into the mucus layer. And so if we look down here at this image at the bottom of the slide, our odorant molecule is this little hexagon here, and it is binding to a receptor protein in the membrane. And again, this receptor protein is embedded in the membrane of these little cilia that make up the dendrites. And these receptor proteins are what we call G-protein coupled receptors. And so when they're activated, there is a second messenger system that happens. We're not going to go into all of the details, but eventually this cascade happens and you eventually open sodium channels. So a sodium channel is closed here, and then after the odorant molecule binds and you get this cascade, the sodium channel opens and sodium rushes in, which causes a depolarizing graded receptor potential. And I also want to point out that in your textbook, you'll often see the term generator potential instead of graded receptor potential, but they are the same thing. So when you see generator potential, you can think graded receptor potential. So as these sodium channels open, you do get this graded potential, and if it's enough to reach threshold, you get action potentials. The action potentials can then be passed down along the axon to the central nervous system. Notice that as far as these little receptor proteins go, you have separate receptor proteins for each kind of smell. So each kind of odorant molecule binds to a specific different receptor protein. And we have at least 50 primary smells, which means 50 different types of these little G protein receptors. And our ability to smell different scents actually comes from a combination, just like if you think about the primary colors of red, yellow, and blue, how you can mix those together. Like you can mix yellow with red to get orange, or you can mix yellow with blue to get green. You can mix these 50 primary smells to be able to sense thousands of different types of smells. And if you think that's amazing, Other animals, like our dogs and cats, actually have more different types of receptors. They have about 200 primary smells. We've had several genes for a lot of our receptors have mutated over our evolutionary history, so we've lost the ability to smell a lot of different chemicals, but a lot of animals like our dogs and cats still have that ability. So if you can think about all the combinations you can make with 50 primary smells, just think about how many more combinations you could make if you had 200 primary smells, and that would give you an idea about the smelling capacity of your dogs and cats. With olfaction, there is very little receptor adaptation, so as long as there are odorant molecules, the receptors will respond and they will generate graded receptor potentials. So there is very little peripheral adaptation that occurs. However, with olfaction, we can get central adaptation, and if you remember from chapter 15, this is where we inhibit nuclei along the pathway. And so with central adaptation, like for example, if you're in a room with a smell you don't like, give it like 15-20 minutes and you won't notice it anymore. That is because of the central adaptation or the inhibition of the pathways getting that information to your consciousness. So speaking of pathways, let's look at the olfactory pathway. So we start off with those olfactory receptors, which are modified neurons that we find at the roof of the nasal cavity, and they're shown over here again in a bunch of different pretty colors. And then these neurons are going to synapse onto neurons in the olfactory bulb, which sits right above the ethmoid bone that forms the roof of your nasal cavity. So the axons from the olfactory receptors will actually pass through the ethmoid bone and then form a synapse onto the olfactory bulb. Note that these olfactory receptors, these modified neurons, are what we call cranial nerve 1, which we covered the cranial nerves last semester. And so last semester we mentioned to you that the cranial nerves, cranial nerve 1, were these short little nerves that go from your top of your nasal cavity up to the olfactory bulb, and now you can better understand that a little bit more. So from the olfactory bulb, the information is going to be passed along the olfactory tract, and then it's going to be split up and it's going to go to several different locations. It's going to go to the olfactory cortex in the temporal lobe. This will allow you conscious perception of the smells. And then signals are also going to be sent to the hypothalamus and the limbic system. Notice that olfaction is the only sense that has a direct connection to a primary cortex, meaning it is not filtered through the thalamus. If you remember from chapter 15, all of your general senses, before they could go to the primary somatosensory cortex, they had to pass through the thalamus. And you're going to see later in this chapter for your other special senses, they also all pass through the thalamus. Olfaction is the only sense we have that goes directly from the receptor straight to the primary cortical area without being filtered by the thalamus. And the connections that our olfactory information has with the limbic system and the hypothalamus also leads to odors being able to trigger very strong memories. So sometimes odors can trigger memory like no other type of stimulus can. And odors can also trigger emotional and behavioral responses due to their connection to the hypothalamus. Like, for example, it can make you feel sad or happy. It can make you start drooling if it's a really good, tasty smell and you're hungry. So it's because of these multiple connections that we can have such strong reactions to odors. And that is olfaction. In this section, we're going to look at your sense of gustation, which is your sense of taste. So let's start by looking at the anatomy of the tongue. So the tongue has little epithelial projections all over it, and these little projections are called lingual papillae. And just as a reminder, when you see papilla, with just an A on the end, that is singular, and when you see the A and the E on the end, like papillae, that is plural. So there are 4 types of lingual papillae. The first one is the filiform papillae, and those are shown down here at the bottom. These can be found on the anterior 2/3 of the tongue, and they run parallel to the midline groove. These type of papillae have no taste buds on them. Instead, they function to increase friction of the tongue for food processing, which we'll talk more about in chapter 28. Then you have the fungiform papillae. These are scattered all over the tongue, but they're especially concentrated on the tip and the sides. Each one of these type of papilla have 5 or about 5 taste buds on it. Then there are the valate papillae. These appear as an inverted V on the posterior side of the tongue, and that's shown in the darker purple circles here. So you have very few of these, only up to about 12 in total. Each one of these, though, has a lot of taste buds, so each one of the valate papilla can have up to 100 taste buds on it. And then the last type is called the foliate papillae, and these are folds that are found on the lateral margins of the tongue, and the taste buds on these are embedded within the folds. So these are epithelial projections, or projections of the epithelial lining of the tongue. And then, as I mentioned, 3 of them have taste buds on them. So let's look at a taste bud in more detail. So again, we have taste buds on our valate papilla, foliate papilla, and fungiform papilla, but not on the filiform papillae. So if we take a single little taste bud, like shown right here in this image, and we look at it close up, as shown up here, we can see that it is made up of gustatory epithelial cells, which are just labeled gustatory cell in this top image. You have about 40 to 100 of these per taste bud. So in this image up here, they are shown as the flat, thin, pink cells. Each one of the gustatory epithelial cells has tiny little microvilli that extend off of the end. This would be the end that goes and faces the environment inside the mouth. And so these little microvilli are often called taste hairs, and they extend into a little depression called a taste pore. Gustatory cells take quite a beating because we're all the time, eating various foods of different consistencies, and it can abrade our tongue. So a typical gustatory epithelial cell only survives about 10 days, so they constantly need to be replaced. So a taste bud also has a supply of basal epithelial cells, and that is shown in this image up here as these darker light brown colored cells that are circular back here. These act as stem cells, so they start out as this basal cell, and then as they grow, they become a transitional cell, which is shown in yellow, and then they mature into a fully functioning gustatory cell, which has those little microvilli on it. And then the actual receptors that do the sensation of the taste that we're going to talk about in the next slide would then be embedded within the taste hairs or the microvilli. So when you're born and you grow up, you have about 10,000 taste buds to start on your tongue as a whole, but the numbers do decline as you get older. So you have a higher sensitivity to taste when you're really young. This is actually why a lot of children don't like a lot of foods, because they taste quite strong to them. And then as we age, we might start eating things that we wouldn't eat when we were a child because it no longer tastes as strong. And then elderly people sometimes have a complaint that all of their food tastes bland because they have lost a significant number of taste buds. There are 6 types of taste receptors. We have sour, which can sense the presence of acids in our food. Bitter, which can frequently sense the types of alkaloids that plants make to warn us that they're poisonous. There are salty, which comes from salt, and then sweet, which comes from sugary foods. Umami is a taste that's kind of like, it's described as being a meaty taste, and it actually, the umami receptors are stimulated by glutamate, which is present in high quantities in proteins. So you can think of umami as more of like a protein type taste. And then yes, we also have water receptors. These are not on the tongue, but rather they are in the back of the throat in the pharynx region. So let's look at the process of gustation itself. So unlike the olfactory sense, in which the olfactory receptors themselves were neurons, in gustation the receptor cells are not neurons, but they do form a synapse with a sensory neuron. So if we look up here on the right, the gustatory epithelial cell now is shown in yellow. Here would be the little microvilli, taste hairs, where these would pick up the stimulus, and then if the gustatory cell has a depolarization past the threshold, then it will send a signal to the sensory neuron down here, which would conduct the action potentials onto the central nervous system. There are two functional types of receptors. So we have two pictures down here. So let's look at the first one. So remember that salt, when something tastes salty, you're actually tasting the sodium in it. And when something tastes sour, you're actually tasting the acidic taste that comes from acids, which is essentially you're tasting the hydrogen ions. So for the salt and sour receptors, we actually have leak channels that allow sodium ions and hydrogen ions to pass through. So if you're eating something salty, that's going to put sodium ions into your mouth, and they will pass through the leak channels and taste like salty. And if you eat something sour, which is like an acid, so think of like orange juice or lemon juice or something that has a really sour taste to it, you're actually putting hydrogen ions in your mouth, which will pass through the open channels. In both cases, when the hydrogen and sodium ions pass through the channels, you get a depolarization, which is a signal that this receptor has been stimulated. The three other major types of receptors, and we're not talking about water. Water receptors are actually aquaporins, so we're not going to discuss those in any great detail. But your three other tastes, sweet, bitter, and umami. In these cases, the chemicals bind to a G protein. It's a receptor that's a G protein. This initiates a cascade inside the cell, which will eventually depolarize the cell, which will cause an action potential in the sensory neuron. So for these three taste sensations, you can think that they work very similar to the way that olfaction worked. So if we take a look at the gustatory pathway, the taste buds on your tongue are innervated by cranial nerves 7, 9, and 10. This is your facial nerve, glossopharyngeal nerve, and vagus nerve. And so the information is picked up from those epithelial gustatory cells, and it travels through the cranial nerves to sensory neurons within the cranial nerves to the solitary nucleus in the medulla oblongata, and then it passes up and it gets filtered by the thalamus. Remember, all sensory information except for olfaction is filtered out by the thalamus. And then it goes to the gustatory cortex, which is in the insula. And if you remember from last semester, to see the gustatory cortex, we have to lift up the frontal cortex and the lateral cortex to look at the insula underneath. And while we're looking at this picture, we can also see that the olfactory cortex that is on the temporal lobe that we talked about in the last section. So taste information is not just from your gustatory receptors, however. Your final perception of taste is a combination of somatosensory information, so texture from tactile receptors on your tongue, temperature from thermoreceptors on your tongue, and even pain from nociceptors on your tongue. And all of that somatosensory information is also mixed with olfactory information. So as you chew, some of the molecules in the food you're eating go up the back of your pharynx and actually stimulate olfactory receptors in your nasal cavity as well. And that's factored into your perception of taste. And if we go back to the somatosensory information, if you've ever eaten those jelly beans that taste like something else, you can tell that the taste is there, but the texture feels wrong, right? You're like, well, this tastes like popcorn, but it doesn't feel like popcorn. It kind of like feels wrong to you. And then I also want to point out that like when you eat hot peppers, like anything that has like cayenne pepper or spicy chili peppers in it, the capsaicin in those dishes is actually activating pain receptors on your tongue. Some people like that sensation and some people don't. But anyway, your overall perception of taste is a combination of what's coming from the gustatory cells plus the somatosensory information plus the olfactory information. And in fact, what we perceive as taste is actually 80 to 90% smell. This is why sometimes, kids will hold their nose when they're trying to eat something they don't like, because if you hold your nose, you're taking away 80 to 90% of the taste that normally comes from olfactory information. This is also why things don't taste as good when you have a cold and you have a blocked nose, because your olfactory information is actually contributing most to your taste sensations. And if you think about it, it makes sense. We only have 5 different taste buds, excluding water. We've got the sweet, sour, bitter, salty, and umami, but we have 50 primary smells. So our wide range of taste that we get, a lot of that is actually due to smell information. We are also more sensitive to unpleasant stimuli than to pleasant stimuli, and by sensitivity I mean the minimum number of molecules required to activate a receptor. And why do you think that might be? And that's because the unpleasant stimuli usually indicate to us that something is bad and we shouldn't be eating it. So like if something is very, very acidic, we want to be able to sense that before the acid can actually damage the membranes in our mouth. If something is very, very bitter, that usually means it has a toxic compound in it, so we want to be able to say, oh, I shouldn't eat this, right? And the same thing if something is off or has gone bad, we want to be able to taste that at very small concentrations before we eat enough to make ourselves sick. And that is the end of gustation. So in this section, we're going to start with the information about our vision, but I'm actually going to break this down into some subsections to make it easier for you to navigate when you're going back and reviewing the video. So we're going to start with a section on the accessory structures of the eye. So the accessory structures include the eyelids, the superficial eye epithelium, and what we call the lacrimal apparatus, which is involved with tear production and drainage. So if we start with the eyelids, they're also called palpebrae, and these are just continuations of the skin around your eyes. The palpebral fissure is the fissure that separates the upper lid from the lower lid, so it would be this gap between the two lids. You also have lateral and medial angles where your upper and lower lids come together. The medial angle would be next to your nose on each side, and the lateral angle would be the side facing closest toward your ear. And the medial angle is also sometimes called the medial canthus, and the lateral angle is sometimes called the lateral canthus. Our eyelids allow us to blink, which allows us to keep our eye lubricated and also act sort of like windshield wipers and removing dust and debris from our eye. And then of course when we're sleeping, keeping our eyes closed prevent the epithelium lining the eye from drying out. We have eyelashes to help prevent foreign matter from reaching the surface of the eye. And then if we take a closer look at the eyelids, we have some glands to talk about. So there are first some glands called tarsal glands, and they also used to be referred to as meibomian glands. And these glands are small sebaceous glands, which are oil glands along the inner margin of the lid. So if we take a closer look at one of your eyelids, so here are your eyelashes coming out down here at the bottom. So sort of behind your eyelids, or behind your eyelashes on the underneath surface of your lid is where you have these tarsal glands. And so they're going to release this oily solution sort of right behind where your eyelashes are coming out. So if you look at this picture here, it kind of shows that you've got all of these tarsal glands. Again, they used to be called meibomian glands, and they are all secreting this oily substance behind your eyelashes. This secretion pretty much helps keep your eyelids from sticking together, so when you blink, your two eyelids don't like just become stuck to each other, and so you get a flawless blinking without even thinking about it. A condition called a chalazion is when you actually have one of these blocked tarsal glands, so the sebaceous secretion can't come out, and so you end up getting a small cyst. And it's called a chalazion when it is not infected, and then if it is infected, this is called an internal sty, and this is like a little infection. Usually you'll have like a yellow dot, and this will be underneath your eyelid. This would be an infection of a tarsal gland. But you also have large sebaceous glands at the base of your eyelashes, and if these become infected, these are called external styes, and these are usually quite visible. They're also quite painful, and so this is an infection of one of those sebaceous glands. In the corner of your eye, in the medial angle, and again, this used to also be called the medial canthus, you have this soft tissue, right? This is that little bit of red soft tissue that you can see in the inside corner of your eye. This area is called the lacrimal caruncle, and this contains some glands as well. And the glands here also secrete various substances, and these substances actually contribute to those gritty deposits that you feel on the inside corner of your eye when you wake up in the morning. The conjunctiva is the name of the epithelium that covers the inner eyelids and the outer surface of the eyeball. So we can divide it into two sections. There's the palpebral conjunctiva, which covers the inner surfaces of the eyelid. So if you lift up your eyelid and look underneath it, you would see the palpebral conjunctiva. And then you also have a bulbar conjunctiva, which covers the anterior surface of the eyeball itself, although it only extends up to the edge of the structure called the cornea, which we're going to talk about a little bit later. And then there is a very, very fine epithelium that does continue on top of the cornea, but it is very thin so as not to interfere with light passing into the eye. Conjunctivitis is a name of the inflammation of the conjunctiva, and this is more commonly referred to as pink eye. So the lacrimal apparatus is the name that we give to a group of structures that are responsible for producing, distributing, and removing tears. So first, why do we have tears? Tears help to reduce friction so that our eyelids can move up and down without abrading against the surface of the eye. They also help to remove debris, so if something gets into your eye, you'll have an increased production in your tears to help wash it out. It's kind of like your own personal eye bath. Tears also can prevent bacterial infection, so they can contain antibodies, which we'll talk more about in the immune system chapter, and then they also contain enzymes that are antibacterial, and these are called lysozymes. And then finally, tears also provide nutrients and oxygen to the outer epithelial layers of the eye, so the conjunctiva. If you remember from last semester, epithelia is avascular, meaning it doesn't have blood vessels to bring nutrients and oxygen to it. It normally gets this from underlying connective tissue, but in the case of the eye, there's not a lot of space for underlying connective tissue, so the nutrients and oxygen are coming from the tears. Also, did you know that tears contain cortisol, which is a stress hormone we'll talk about in chapter 18? But when you cry, you're actually releasing cortisol as well as some other waste products from your body. And this is why having a good cry can actually reduce your stress levels and make you feel better afterwards. So it's okay to cry, whether you're a man, woman, or anything in between. Feel free to cry when the need is there. So let's look in more detail at the lacrimal apparatus. So tears are produced by what's called the lacrimal gland, which is an almond-shaped gland at the lateral upper corner of each eye. So you have two lacrimal glands, one on each side. The lacrimal glands produce the largest volume of your tears. Each gland makes approximately 1 milliliters of tears per day. Now that is if you don't cry. If you actually have a crying fit, they're going to produce a lot more. When the tears produced by the lacrimal gland mix with the sebaceous secretions by those tarsal glands and the other accessory glands we mentioned, that forms sort of like an oil slick type of consistency, and this allows the eyes to remain lubricated, and it also slows evaporation of moisture from the eye. So now let's look at the other structures involved in the lacrimal apparatus, and we're going to do that by looking at the pathway of tear production and elimination. So the tears, as we mentioned, are produced by the lacrimal gland up here in the upper corner of the eye. Then they're going to pass through these small little lacrimal gland ducts where they're going to be released onto the ocular surface. They're then spread around your eye by blinking. And then eventually they make their ways to the medial angle of the eye, which we also call the medial canthus, and they're going to cover the lacrimal caruncle in this eye. And oftentimes tears can build up in this location, and it looks like a little lake of tears in the corner of your eye. Then in this region there are two very small holes called lacrimal puncta or pores. A single one is called a punctum, and together they're called puncta. So the lacrimal puncta or pores is where the tears will drain. So they'll drain into one of each of those two holes. If you look very closely at the medial angle of your eye in the mirror, you might be able to see one or both of these little holes. After it passes through these holes, it's going to pass through these two little canals here. These are called lacrimal canaliculi. A single one would be a canaliculus. So you have a superior lacrimal canaliculus and an inferior lacrimal canaliculus. From there they pass into a structure called the lacrimal sac, and then from there they're going to move downward through this nasolacrimal duct, and then they empty out through an opening into your nasal cavity. And so because tears drain from your eye to your nasal cavity, this is why when you do cry you end up with a runny nose. That's actually not snot coming out of your nose, it is your tears. In this section, we're going to look at the eyeball anatomy, and so we're going to look at how the eye can be divided into 3 layers and what the major structures are of each layer. So first we can divide the eye into 3 layers, and these used to be called tunics. So the first one is the outermost fibrous layer, and that is shown here in this white and gray in this image, and that is composed of the cornea and the sclera. The middle layer is called the vascular layer or the uvea, and this is shown in this image in this sort of yellowish beigey color, and it's made up of the iris, ciliary body, and choroid. And then we have the inner layer, which is made up of the neural layer and pigmented layer, and this is also called the retina. And then here is another image that is color-coded to help you see the difference between the 3 layers. So the fibrous layer here would be the outside components, the vascular layer would be the middle components, and then the retina or the inner layer would be the deepest components. So let's look at each of these. So we'll start with the fibrous layer. So remember, the fibrous layer is composed of the cornea and sclera, and it functions to support and protect the eye, to serve as an attachment site for the extrinsic eye muscles, and it also helps to assist with the focusing process. So if we look at just the cornea, this is that transparent portion on the very anterior surface of the eye. It's transparent to allow light to pass through, and it's made up of a dense matrix of collagen fibers that are arranged so that they don't interfere with the passage of light. And as I mentioned before, it also has a very thin epithelial layer that surrounds it. There are no blood vessels in the cornea because they would interfere with the passage of light. So for that reason, as we'll see in a moment, the cornea can't heal very well. But it does have numerous free nerve endings, and so the cornea is the most sensitive part of the eye. So anything that touches your cornea is going to cause a reflex of you to blink your eye or move away. Also, if you get a scratch or damage to the cornea, it can be very, very painful for many days. So again, because it doesn't have blood vessels, the cornea has a very restricted ability to repair itself. So if you do have major corneal damage that results in scarring on the cornea, this can interfere with your vision. And so a corneal transplant is actually one of the most common types of transplants, and you can donate your corneas by indicating so on like your driver's license as you being an organ donor. The cornea specifically provides the initial refraction or focusing of the light, and we'll look at that in a little bit more detail when we talk about focusing. And then the sclera, which is shown in this purple color, this is known as the white of the eye. So this is the largest portion of the outer part of the eyeball. And it is composed of a very dense fibrous connective tissue. And when you're in lab, you'll see just how dense it is. It's actually pretty difficult to cut through. And the sclera is going to provide an attachment site for the 6 extrinsic eye muscles that move your eyeball up, down, sideways, and diagonally. And you'll go over those 6 extrinsic eye muscles in lab as well. So next we have the vascular layer, or the uvea, and this is made up of the iris, the ciliary body, and the choroid. So overall, the vascular layer functions as a route for blood vessels and lymphatic vessels that supply the tissues of the eye. It also regulates the amount of light that enters the eye. It secretes and absorbs aqueous humor, and it controls the shape of the lens, which is necessary for focusing. So if we look at these parts individually, the iris is the highly vascular pigmented tissue that we all associate with the color of our eye. And the iris itself is actually composed of 2 sets of muscles, and these are smooth muscles that can change the size of the pupil. And the pupil is that hole in the middle through which light actually enters the eye. So again, if we take away all the pretty colors of the iris and just looked underneath the colors, we would see that there are 2 sets of muscles. There are radial pupillary dilator muscles called the dilator pupillae, and when they contract, this is going to dilate the pupil or make it larger to allow in more light. And then we also have concentric pupillary constrictor muscles that are right around the pupil in a circle there. Those are called sphincter pupillae, and when they contract, it makes the size of the pupil smaller, which allows less light to pass through. And then the iris itself is also very highly pigmented. The color comes from the presence of melanin, which is the same pigment that determines your skin color. So the darker your eye color, the more melanin you have. And blue eyes actually represents the least amount of melanin possible. So if we take a look at the ciliary body, the ciliary body is located here on either side of the lens. And the ciliary body has a muscle inside of it called the ciliary muscle, which can pull on the lens and change the shape of the lens. And as we're going to see later, this helps with the focusing, and we'll go into more detail on how light is focused in another section. The ciliary zonule, also called the suspensory ligaments, are these little small ligaments that stretch from the ciliary body itself to the lens, and so they hold the lens in place to make sure that it stays right behind the pupil. So light's going to pass through the cornea, then through the pupil, then through the lens. The ciliary body also secretes aqueous humor, which we're going to discuss in the section on the chambers of the eye. And the last structure of the vascular layer is the choroid. This is a middle vascular layer that has a very extensive capillary network. And so you can see it in this picture up here is this red portion. It's between the white sclera and the yellow of the retina. So this would be the choroid all the way around the middle of the eye. It delivers oxygen and nutrients to the cells that make up the retina. And it also contains melanocytes, which are cells that produce the pigment melanin. And our last layer is the inner layer, also called the retina. And this one has one primary function, and that is photoreception, which we'll cover in detail in another section. It's composed of two layers. There is the neural layer, which is shown up here with the rods and the cones, and then these other neurons called ganglion cells and bipolar cells. So it is pretty much all of this area right here. So the neural layer contains photoreceptors, supporting cells, and neurons that are going to perform the preliminary processing and integration of visual information. And we're going to go a lot more into detail on photoreception and how this works in later sections. For now, I'll just tell you that a term to be familiar with is retinopathy, which is any disease of the retina. There is also a pigmented layer, which are these little square-shaped cells at the very back here. These are also called the retinal pigment epithelium. And these little cells contain pigments that absorb light or photons that somehow make it through the neural layer without hitting any of these cells and without being absorbed by any of the cells of the neural layer. And so the pigmented cells back here serve to catch those stray photons of light so that they don't bounce back and hit the photoreceptor on their way back out, because that would prevent visual echoes, and we don't want that. We want nice, crisp, clear, sharp vision. So they are there to catch those stray photons of light. And again, we'll go in more detail in another section. In this section, we're going to talk about the chambers of the eye and differentiate between aqueous and vitreous humor. So the eye is actually a hollow and fluid-filled cavity. So there are a couple of cavities within the eye that are filled with fluid. And the first one is the anterior cavity, which is shown in this light purple color up here. The anterior cavity lies between the cornea and the lens, and this part contains aqueous humor, which is a very watery-based fluid that we'll talk about in more detail in a moment. The anterior cavity itself can be divided into an anterior and posterior chamber. So be careful when you use the term cavity and when you use the term chamber. So the anterior cavity has an anterior chamber in front of the iris, and then behind the iris it is a posterior chamber. So the iris is the dividing line within the anterior cavity. There is also this large posterior cavity which sits behind the lens, and this makes up the main body of the eyeball. It does contain some aqueous humor, but the majority of the space in the posterior cavity is taken up by what's called the vitreous body, which is actually a gel-like body that when you do your eye dissection in the lab, you can see looks like a piece of jello. There is a small fluid component to the vitreous body called vitreous humor, but the majority of it is this gelatinous substance which helps to stabilize eye shape. You are born with all of the vitreous body and vitreous humor that you're ever going to have. However, your aqueous humor is constantly being made and replenished and recycled, as we'll see in just a second. So let's take a closer look at the aqueous humor. And we're also taking a closer look up here at the anterior cavity, which again, remember, can be divided into an anterior chamber in front of the iris and a posterior chamber behind the iris. So aqueous humor itself is formed by epithelial cells within the ciliary body. So you can see the little red arrows here represent the production of the aqueous humor. It's then secreted into this posterior chamber of the anterior cavity, and then you can see this little area here is going to go through the pupil. It's going to pass around to the anterior chamber of the anterior cavity, and then it's eventually going to leave through this opening called the scleral venous sinus, where it's going to go back into the bloodstream through the venous system. So this is very similar to how we talked about the production and reabsorption of cerebrospinal fluid last semester. There is also a small amount that enters the posterior cavity, but remember the largest majority of the posterior cavity is taken up by the vitreous body. Aqueous humor actually resembles cerebrospinal fluid in its composition, and it's important for nutrient and waste transport for many of the cells and structures within the eye, like the retina, and structures within the iris itself and the ciliary body, just multiple structures within the eye. It also helps to form a fluid cushion, which allows the eye to absorb some shock and impact. Because we have fluid in our eye, we can measure what is called an intraocular pressure, which is the pressure of the fluid inside the eye. And when you go to the eye doctor, they usually measure this at the level of the cornea. So they measure the amount of pressure of the fluid pushing up against the cornea. The reason why it's important to have your intraocular pressure monitored is because if it is too high, it actually has nowhere to go because of the thick fibrous outer layer of the eye. So the only place the extra fluid can find a way to sort of escape the confines of the cavities is to push out against the optic nerve. And this is called glaucoma, when you have increased intraocular pressure that pushes against the optic nerve and disrupts your sight and can lead to permanent blindness. So why do we need intraocular pressure, and why do we need the fluid inside these eyes? It's not just to provide the nutrients and the waste transport. The pressure also holds the retina in place up against the pigmented layer in the choroid. So when you're in lab, you'll notice the retina is not actually attached to the choroid behind it. It's kind of floating free. And so without the intraocular pressure of the fluid, the retina would actually fall off the back of the eye, and that's what we call a detached retina. So you need just the right amount of pressure, not too much or you'll get glaucoma, and not too little or you can have a detached retina. In this section, we'll look at the focusing of light, and then we'll talk about visual acuity. So first, a quick review of what light is. So a photon is the basic unit of light. So light can act as both a wave and a particle. So we call a single packet of light a photon. And light energy is a form of radiant energy because it travels in waves. And each photon of light has a characteristic wavelength. So we typically talk about the electromagnetic spectrum of different wavelengths. And what we call visible light are these wavelengths shown at the bottom, and these are the wavelengths that the photoreceptors in our eye can pick up, which is why they are visible. There are other animals in the animal kingdom that have receptors in their eyes that let them pick up other parts of the electromagnetic spectrum. For example, birds and bees can see in the ultraviolet range, which we cannot. So the visible light spectrum refers to what humans can see with our photoreceptors. And we have two types of photoreceptors. We're going to go into more detail later, but just as an introduction, we have a type called rods, which tells us about the presence or absence of photons, so whether light is present or not. And then we have a type called cones, which actually tells us about the specific wavelength of a particular photon, and this allows us to see colors. So light waves are refracted, which means they're bent when they pass from one medium to another, so from like air to liquid or liquid to solid. And you can see this like if you stick a pencil in a glass of water. So as the light waves are traveling from the air, and then they have to travel through the water, so there's the change from one medium to another, you can actually see the refraction of the light. And you can see this too if you shine a light through a prism, like a piece of glass. You can see the light bends as it enters the solid and then bends again when it goes back to the air. So in our eyes, the largest amount of refraction occurs as the light goes from the air into our cornea. But we also have additional refraction when the light passes through the aqueous humor in the anterior cavity as well as the lens. And then our lens is responsible for refracting the light in such a way as to allow the waves of light to bend so they fall on a precise focal point on the retina, and we call that focusing. So the lens is responsible for focusing the arriving image, and the lens is formed from fibers that have transparent proteins called crystallins. Crystallins are not renewed throughout your life, so the crystallins that are part of your lens when you're born are the ones that you'll be stuck with. And a condition called cataract occurs when the lens loses its transparency. So over time, aging can cause accumulated damage to those crystalline proteins, and they can turn yellowish and cloudy. And so with a cataract, the lens becomes clouded, and so light no longer, or images no longer look as sharp and focused because the clouding of the lens causes the light to scatter. And so here's a picture of a normal eye, and if you look in the pupil, because remember the lens sits behind the iris, that's where you can see the cloudy lens. And senile cataracts, which are cataracts that happen because of aging, those are the most common, but cataracts can also be caused by injuries to the eye, UV radiation, or a reaction to certain medications. And UV radiation actually contributes a lot to the development of cataracts as you age. So while you're young, make sure you're wearing sunglasses when you're out in a bright sunny day, because the UV radiation can slowly damage your lens. So if we look at the path of light through the eye, so the light comes in through the air, and then it's going to pass through the cornea, then it's going to pass through the aqueous humor in the anterior cavity, then it's going to go through the pupil, which is the opening in the iris, then it's going to pass through the lens, then it's going to pass through the posterior cavity filled with the vitreous body, and then it's going to be focused onto the retina. So we have that initial large refraction of light as it passes from the air to the cornea. There is also some refraction as it passes through the aqueous humor of the anterior cavity. The lens does the purposeful refraction, so it bends the light in order to focus it onto the retina. And then there is a small amount of refraction that also occurs as it passes through the vitreous body in the posterior cavity. Note that the iris can adjust the size of the pupil to adjust the amount of light coming in. So if the pupil dilates, it allows more light to come in. If the pupil constricts, it allows less light to come in. And note that the ciliary body is what is actually adjusting the shape of the lens to help with the focusing process. So we use the term accommodation to describe the automatic adjustment of our eyes to focus an image. And again, the lens is providing that extra refraction needed to focus the light onto the retina. The focal point is that specific point of intersection where that final image hits the retina. And the lens can adjust the focal point by altering its shape. So when the ciliary body pulls on the lens, it becomes more round or flat. So the greatest amount of correction is needed if you look at something very close up. So when you're looking at something very close up, the ciliary muscles have to contract, which pulls on the lens and makes it more rounded. Because when you're looking at close-up objects, you have to have the contraction of the ciliary muscle. Your eyes can tire if you use the computer, phone, or if you're reading a book for too long. In the case of a computer and phone, we actually call this digital eye strain. So it's a good idea to look up at a point far away every so often to rest your eyes. And also, because these muscles wear out over time, the older you get, the harder it is to focus on really close objects, which is why as you age, many people need to get reading glasses. For distant vision, the ciliary muscles relax and the lens becomes more flattened. And so this doesn't require any active muscle contraction, which is why you can rest your eyes by looking at something far off in the distance. So some people are born with accommodation problems. So in the condition called myopia or nearsightedness, the eyeball can either be too deep or the curvature of the lens can be too great. And so the focal point of the image falls in front of the retina. So that means that the person is going to see objects in the distance as being blurry, but looking at things close up are okay. So they can see near, but they can't see far. So you can correct this with lenses, either glasses or contacts. In the condition of hyperopia or farsightedness, the eyeball is either too shallow or the lens is too flat. Again, you can be born with these conditions. And in hyperopia, the focal point is going behind the retina. So you can see things clearly at a distance, but you have trouble focusing on things close up. So this is why it's called farsightedness, because you can see far, but you have trouble seeing close up. And again, you can correct this with glasses and contacts. There's another common condition called astigmatism. In astigmatism, different areas of the lens focuses the light differently. So this results in more than one focal point on the retina. So with astigmatism, you might have like a copy of the image because you're actually looking at multiple focal points instead of just the one single focal point that you should have. Also note that when the image arrives on the retina, it is upside down and the left and right are reversed. So for example, if you're looking at something high up on your wall, that is actually going to hit the bottom of your retina, and vice versa. A point low on your wall will hit the top of your retina. And then left and right are reversed as well. So like if you see the blue fence post here, will turn out, if it's on the left of your vision, it'll turn out on the right side of your retina, and vice versa. So you're never aware of this because the brain compensates so that you see everything up right side up and correctly oriented. But this does become important, like if you're an eye doctor and you need to know what part of the retina is damaged, this can help you figure it out. And then the term visual acuity refers to how well you see based on a comparison to a normal standard. So what we call 20/20 vision is when you can see at 20 feet what a normal person can see at 20 feet. So basically you're the same as a normal person. And I find that students can sometimes have a difficulty understanding visual acuity, so I tried to draw a little graph for you, or a little illustration. So this is you and a normal person. You're standing side by side, so you can both equally see stuff from 20 feet away, and that's what we call 20/20. With 20/15 vision, your little green stick figure, you can see something 20 feet away that it takes a normal person has to be 15 feet away. So you actually have better vision. You are able to see the details from farther away than a normal person. So that would be 20/15. So notice the first number is always referencing you, and the second number is always referencing the normal person. So with 20/30 vision, you can see at 20 feet what a normal person can see at 30 feet. So now you have to be closer to the object, so your vision is not as good. You can't see as much detail from as far away, so this would mean that you have worse than normal vision. Visual acuity of 20/200 is when a person is considered to be legally blind and like they wouldn't be able to get a driver's license. And so to illustrate this, you would have to be 20 feet away to see something that a normal person could see from 200 feet away. So basically you have to be right up on it. If you go any farther than this, you can no longer see it. True blindness is a total absence of vision. So the 20/200 people can still see, at most everything's blurry, but you're considered to be legally blind. Like I said, you can't get like a driver's license and stuff. But total blindness is when your vision is totally gone. This can be caused by diabetes, which can damage the retina and cause diabetic retinopathy. It can be caused by cataracts that are left untreated. It can be caused by glaucoma that damages the optic nerve. Can be caused by corneal scarring that scratches the cornea so that light no longer can enter it. Can be caused by a detached retina that comes off the back wall of the eye. Also just other types of injuries and even genetics. In this section, we're going to look at a closer look at the retina and the organization of the cells in the retina, and then talk about the two different photoreceptors. So remember, the retina is composed of a neural layer and a pigmented layer. The pigmented layer we've already talked about, and that's going to be this layer right up here that is right up against the choroid. So now we're going to focus on the organization of the neural layer, which contains the photoreceptors themselves and the supporting cells. So there are two types of photoreceptors, rods and cones, and we'll go over the difference between those in a moment. There are more rods around the periphery of the retina, and as you get to the center of the retina, there are fewer rods and more cones. So the macula, which is shown by this dotted line in this image, is a region containing only cones, so there are no rods in the macula. In the condition called macular degeneration, you have problems with this area of the retina, and so you lose your center vision. You can still see things on your periphery, but you can no longer see things right in the very center area of your vision. And then the very center of the macula is a region called the fovea centralis. This is where you're going to find the highest concentration of cones on the entire retina, and so this is the area of your sharpest vision. The focal point that we talked about in an earlier section should fall onto the fovea centralis for your clearest and sharpest vision. Also, just to note, to get a good look of your retina like this, to check the structural soundness of it, this is why your eye doctor always wants to dilate your eyes. So if we look at the neural layer in more detail, the photoreceptors are up here next to the pigmented layer. We have rods and cones that we'll talk about in a moment. The photoreceptors are then going to synapse onto bipolar cells, which are these little red cells up here in this image. So bipolar means they have two extensions coming off of their cell body. The bipolar cells are going to form a synapse with these ganglion cells down here at the bottom. And then all of the axons from the ganglion cells, so you see how all the axons come together, they come down here and they connect. These are all going to come together at a central point to form the optic nerve. So if we're looking at the flow of information through the retina, the photoreceptors are the ones that sense the light, and then they send a signal to the bipolar cells, who send a signal to the ganglion cells, and all of the axons of the ganglion cells then come together to make up the optic nerve, which is cranial nerve number 2. Note that there are some other cells involved. There are these cells called horizontal cells, shown in purpley color, and amacrine cells, shown in brown. These cells can alter the communication of this network. They can inhibit and facilitate signals. And it's really interesting in how they do this and how it plays a role into our vision, but it's beyond the scope of this course. And then, as I mentioned, all of the axons of those ganglion cells come together to make up the optic nerve. And then the place where those axons dive through the back of the eyeball to go to the brain, blood vessels also come in and out of this point. This place where the optic nerve and the blood vessels leave the back of the eye is called the optic disc. The optic disc is a blind spot. Because we have all these axons and blood vessels in this area, there are no photoreceptors here. So see the photoreceptors right up against the pigmented layer? Right in the place where you have the optic disc, there are no photoreceptors. Therefore, any light that falls on this area will not be able to be sensed. Therefore, it is a blind spot. But our brain fills in that missing information, so we're never aware of the blind spot unless we do those little activities they designed to help you actually see your blind spot. So let's talk in more detail about the photoreceptors. So photoreceptors are highly specialized receptor cells for detecting photons of light. Photoreceptors of both types have an outer segment, and the outer segment consists of flattened membranous discs that contain visual pigments that do the actual light detection. In the rods, which are shown on the left here, you can see that the outer segment looks like a little rod, hence the name, and the discs are stacked up on top of each other. Each one is separate from the other. In the cones, shown here on left in purple, they're so called because their outer segment comes to a blunt point, so it looks like a cone, and their discs are actually these infoldings of membrane that go back and forth, so each disc is not separate from the others. When you look in these infoldings of membrane in the outer segment, this is where you will find visual pigment molecules, and the visual pigment molecules are the ones that actually will absorb the photons of light. And the pigment molecules are composed of two main parts. There is a protein shown in purple over here that looks a bit like a dumbbell called an opsin. So that's the name of that overall protein. And then in the middle of the opsin is a molecule called retinol. And retinol is derived from vitamin A, and vitamin A is high in foods like carrots, which is why you've probably grown up hearing that carrots are good for your eyes. Now, there are different types of these overall pigment molecules in rods and cones. In rods, it's called a rhodopsin molecule. So the specific type of opsin used is called rhodopsin. And then in cones, there are other types of opsins. So that's how they're different. They've got slightly different pigment molecules. The inner segment of the photoreceptors are just your normal cells with the nucleus and Golgi apparatus and mitochondria and rough ER. So they have the basic cell anatomy that we talked about in A&P1. And then note that the photoreceptors, both types, are going to form a synapse with a bipolar cell. So we looked at that pathway on the previous slide. Photoreceptors talk to bipolar cells. So let's take a look and compare rods with cones. So if we look at the shape of the outer segment, rods have that elongated cylinder that looks like a little rod, and the cones have an end that tapers to a blunt point, making them look like a cone. The membranous discs of the rods, each one is a separate entity, whereas with the cones, it's a continuous infolding of the plasma membrane. The visual pigment molecule in rods is called rhodopsin. In cones, there are 3 different opsins that detect light at different wavelengths. So let's take a look at this little graph down here. So you can see rhodopsin is shown here where it says rods. That is the primary wavelength that is absorbed by the rhodopsin molecule. And then we have 3 different opsins. They're called blue cones, red cones, and green cones based on their area of the visual light spectrum that they can detect. So blue cones detect light in the blue wavelength areas, red cones more toward the red end, green cones more toward the middle. If you've heard of someone being colorblind, that means they have a mutation in one or more of these 3 opsin genes. The most common type of color blindness is red-green color blindness, in which there is a mutation in either the red opsin or the green opsin. And if you have red-green color blindness, you could not see the number 74 here that is in green on a red background. It would just look all the same to you. And then if we look at the functional characteristics, rods function best in low light. They are best at showing the presence or the absence of light. So images that you look at using just your rods appear black and white. And rods are the photoreceptors we use the most, like at night if we're walking around the house in the dark, or outside when it's very low light, like when the sun's setting. All of that type of vision is our rods. Cones function best in bright light, so like the middle of the day, bright sun, and they allow us to differentiate between different wavelengths of color and provide us with our color vision. In this section, we'll look at the process of photoreception. So this is how we convert a photon of light to an electrical signal. So first, I must explain what's going on inside the photoreceptors at rest. And by at rest, we mean in total darkness, where there are no photons of light around. So at rest, if we take a closer look at these little infoldings of membrane in the outer segment of the photoreceptor, we can see that at rest, there are sodium channels that are held open by a little molecule called cyclic GMP or cGMP. So cGMP is around in the dark and it binds to the sodium channels and it keeps them open. Well, if you remember from last semester, an open sodium channel means sodium is going to rush into the cell. So at rest, we actually have sodium actively rushing into the photoreceptor, and we call this a dark current because it's taking place in the darkness or in the absence of light. So having this active sodium coming into the cell keeps the photoreceptor depolarized. And so remember, resting potential is normally negative 70 millivolts. And so at rest, the photoreceptor, because it has the sodium coming in, actually has a membrane potential of minus 40 millivolts, which is depolarized above threshold. So it's actually sending action potentials or signals through neurotransmitter release to the bipolar cell. So the bottom line, and this is a little backwards from kind of the way that you learned things before, but the bottom line to remember and to get in your brain is that if you have an active signal from a photoreceptor, meaning that the cell is depolarized and sending a signal through neurotransmitter release to the bipolar cell, that indicates that there is no light around. All right, so in the dark, in the photoreceptor's resting state, there is an active signal from the photoreceptor to the bipolar cell. So now let's look at the process of photoreception. And this is the process of taking a photon of light and converting it into an electrical signal. And there are 4 steps in this process. So we start with the photon of light that arrives into the photoreceptor, and it's going to activate an opsin molecule. So remember, these opsin molecules, rhodopsin is the type that you find in the rods, and then there are 3 other opsins in the cones. All of them have this retinol molecule on the inside, and at normal resting conditions, this retinol has a little kink in the tail, and this is called 11-cis retinol. And so when a photon of light appears, this photon of light hits that retinol molecule and it converts it to 11-trans retinol, which means it makes the tail straight. So the photon of light is converting 11-cis retinol into 11-trans retinol, and this causes the overall opsin protein to change shape, and we then say that the opsin is activated. So step 1 was activation of the opsin by a photon of light. Step 2 is a signal cascade that occurs because of the activated opsin. So we have this activated opsin, and when it changes shape, it's going to turn on or activate an enzyme called transducin, which sits right next to it. It's this little flat protein here. And then when transducin gets activated or turned on, it's going to activate or turn on another protein called phosphodiesterase, abbreviated PDE. So this is the little signal cascade. This means that one molecule turns on another, which turns on another, and it happens in a row. We call that a signal cascade. And PDE, phosphodiesterase, is an enzyme that breaks down cyclic GMP molecules. That becomes important in our next step. But to summarize step 2, we have a signal cascade. The activated opsin activates transducin. The activated transducin activates phosphodiesterase. In step 3, the cyclic GMP levels decline and the sodium channels close. So remember, if we go back to our resting state, we normally have these little cyclic GMP molecules that hold the sodium channels open. Also remember that phosphodiesterase, or PDE, is an enzyme that breaks down cyclic GMP. So because we activated PDE in the previous step, It is now breaking down the cyclic GMP. And after it's breaking down the cyclic GMP, there is nothing left to hold the sodium channels open. And so the sodium channels are going to close. So step 3 was the decline of the cGMP levels, and then sodium channels close as a result. In step 4, the dark current is reduced and the photoreceptor stops signaling. So in the previous step, we just closed the sodium channels. So no more sodium is flowing into the cell. We instead have sodium-potassium pumps that's pumping the sodium back out. So because that sodium current stopped, now the photoreceptor can return to a resting potential of negative 70 millivolts, which is not high enough to hit threshold. So now it is no longer going to be sending a signal to the bipolar cell. So the photoreceptor stops signaling to the bipolar cell. So again, a key point, when the signal stops from the photoreceptor, that is an indication to our central nervous system that there is light present. So it's a little bit backwards than what we typically think of, but this is the way this process evolved for whatever reason. So having a signal from a photoreceptor means there is no light present, and when that signal stops, it means that light is now present. And that is the process of photoreception. In this section, we'll look at bleaching and adaptation, which involves recycling the visual pigments and also light versus dark adaptation. All right, so remember that as part of the pathway for photoreception, we have to activate the opsin molecule by converting the 11-cis retinol to 11-trans retinol. Well, what happens after the opsin has been activated? It actually has to be reset before it can absorb another photon of light. So as long as it stays with the 11-trans retinol on the inside, it cannot absorb another photon of light, so it cannot react a second time. So it has to undergo this process called bleaching, in which the opsin molecule is broken down so that the 11-trans retinol can be converted back to 11-cis retinol, and then the opsin molecule is reassembled. So if we look at this image from your textbook, so we start here with the opsin and the 11-cis retinol, so this would be the starting normal condition. Remember that when a photon of light hits this molecule, it causes the 11-cis retinol to convert to 11-trans retinol. And then we're stuck like this until we undergo bleaching. So the bleaching process is when we first take apart the opsin, we separate it from the retinol, and then an enzyme comes in and converts the 11-trans retinol back to 11-cis retinol, and this does take energy. And then the 11-cis retinol is combined or joined back up with the opsin, and now it is ready to go again, and now it can absorb another photon of light. So this process is called bleaching. So let's look at the two major types of adaptations. One is when we've been in the dark for a while, and the other is when we've been in bright light for a while. So when you first walk into a dark room, right, you might not be able to see much, if anything at all. But after you have stayed for some time in that dark room or outside in the dark, almost all of your visual pigments, so all of those opsin molecules, have recovered from bleaching. So all of those 11-trans retinols have been converted back to 11-cis retinols, and now they are fully receptive to new photons of light. And the reason they have time to do this is because in the dark, there are very few photons of light around to keep activating them, so it gives them time for them all to reset. So in the dark, after you've been in the dark for a while, your visual system becomes extremely sensitive because all of those opsins are ready. They're all holding an 11-cis retinol, and they're all ready to absorb a new photon of light. So when we say that it's extremely sensitive, this means that a single photon of light is able to hyperpolarize like a single rod and send a signal to the brain. So you can imagine if you were to look at a little cross section of the retina, and these are all of your rods and cones back here. So after you've been in the dark, when you're dark adapted, almost all of your photoreceptors, they've already undergone the bleaching process, so they're all bleached. They all have 11 cis retinol, and they're all ready to receive a new photon of light. So just a single photon of light can come in, and almost anywhere it hits, it's going to find a photoreceptor that is ready to be stimulated. So like I said, when you first go outside or walk into a dark room, things might not be as clear, but after you've been in the dark for a while, you reach this dark adapted state, and then the smallest amount of lights are visible. So when you go from the dark to the light, first of all, so think about like when you walk outside after seeing a movie and you've been in the dark theater, you have an initial shock because remember, when you were in the dark, all of these pigments were ready to receive photons of light. So when you first walk out of a dark room into the light, you're hit all at once. All of those photons activate all of those photoreceptors, and it can almost be too much. It makes you squint, makes you cover your eyes. And so there's that initial shock of most of the pigments responding at once. But then as you stay in bright light, the rate of bleaching becomes balanced with the rate of reforming, and the whole system has a reduced sensitivity, but because there's still enough photons around, because it's bright light, you're still able to see. So in a light adapted state, a single photon is less likely to find a ready photoreceptor. So think of it like this way. If we look at our little line of photoreceptors again, you know, this guy up here, he's bleached and he's ready for a new photon of light. The guy underneath him, he's undergoing that process of bleaching where the opsin is being broken down, separated from the 11-trans retinol. The enzyme is coming in to convert the 11-trans retinol back to 11-cis retinol. So this particular photoreceptor is not ready for another photon of light. This one's ready, this one's not ready, this one's ready, this one's not ready. So basically, your system reaches an equilibrium where about half of the photoreceptors are ready at any given point and the other half are undergoing the bleaching process. This is what we call the light adapted state. So the whole system has a reduced sensitivity because a single photon of light has a higher chance of hitting one that's not ready, so it won't get activated. However, because you're in a bright light situation, there are enough photons of light around to hit ones that are ready that you're able to see. So we call that light adaptation. And this is the last section for the vision section of this chapter, and we're going to look at the visual information pathway. So we already talked about how information flows through the cells of the retina. So as you recall, the photoreceptors communicate with the bipolar cells, which communicate with the ganglion cells, and then all of the axons of the ganglion cells come together to form the optic nerve. Now let's look at the rest of the pathway and how the information gets to the brain. So the optic nerve, which again is cranial nerve number 2, leaves the back of the eyes, and then it's going to cross over at a place called the optic chiasm. Signals from the medial retina on each side will cross over, whereas signals from the lateral retina on each side will stay on the same side of the body. What this results in is that all of your left visual field from both eyes is going to make it to the right side of the brain, and all of the right visual field from both eyes is going to make it to the left side of the brain. And that is because of the crossing of fibers at the optic chiasm. After the optic chiasm, we call these nerves the optic tracts. Then they are going to go to the thalamus, because remember, all sensory information except for olfaction is processed and relayed through the thalamus. Specifically, visual information goes to an area of the thalamus called the lateral geniculate nucleus, or the LGN. And then from the thalamus, signals are going to travel to the primary visual cortex, which is in the occipital lobe. And there is a map in this cortex that maps out to all of the locations on the retina, as we discussed in chapter 15 when we talked about cortical maps. So visual processing centers of the cortex, so after the information reaches our cortex, are really good at filling in gaps of any missing visual information. This is why we aren't aware of our blind spots. So even if you close one eye, you should have a giant blind spot in the middle of the other one, but you don't, because the brain fills in those missing gaps. And this forms the basis for many of our optical illusions. For example, most people can see the Dalmatian that is walking along sniffing the ground in this image without having to be shown it or to see the outline. If you can't see it, there is an outline there for you to see, but the brain kind of automatically takes this pattern of light and dark and fills in the missing information to make it into a picture. It's also why illustrations like this make us believe there is a triangle there, or we see a triangle, but there actually isn't a triangle there. There are 3 circles that have a pie slice cut out of them, but because of the way they are oriented to each other, our brain wants to fill in the missing information and insist that there's a triangle there. So that was the main pathway of information from the eyes to the visual cortex. However, there are some of the axons from the optic tracts that branch off and go to other locations. One of these locations is the superior colliculus in the midbrain. This is part of the tectum. And this is the integrating center for eye movements. And this area also controls reflexes to visual stimuli. So like when you whip your head around to see something that's moving in the corner of your vision, or when you blink when something's being thrown at you, right? This is the reflex center for visual reflexes. There is also a branch of the optic tract that goes to the suprachiasmatic nucleus of the hypothalamus. This area of the hypothalamus is our master circadian clock that controls our daily and seasonal rhythms. And then the suprachiasmatic nucleus also will inhibit the pineal gland and stop it from producing melatonin when there is light present. So the presence of light, either artificial or natural, will inhibit the production of melatonin. We discussed this last semester, and we're going to talk about it again briefly in chapter 18. And that is it for vision. Now we have 2 senses left. Okay, in this last major section, we're going to look at the senses of equilibrium and hearing together, because they both involve structures inside the ear and they both use the same type of receptor. Again, it's a bigger section, so we are going to break it up into some subsections. So we're going to start with the anatomy of the ear. And the ear can be divided into 3 major regions. We have the external ear, the middle ear, and the inner ear. So we're going to go over the structures in each section separately. So if we start with the external ear, the first major structure that we see is the auricle, which is the elastic flap that makes up your ear flap. It's also called the pinna. The auricle protects the opening to the inner ear area, and it also provides directional sensitivity. So because it interferes with sound waves that, say, like, for example, come from behind you versus sound waves coming from in front of you, because it interrupts those sound waves, it allows your brain to tell the differences so that it knows whether a sound is coming from behind you or in front of you. So we say that it provides directional sensitivity. Then you have what you probably used to call the ear canal, but now you're going to call the external acoustic meatus. This is also sometimes called the auditory canal, but it's better to call it the external acoustic meatus, especially so we don't confuse it with the auditory tube, which is another structure altogether. And the external acoustic meatus provides a passageway for sound waves to get into the internal structures that are responsible for allowing us to hear. There are glands called ceruminous glands within the external acoustic meatus, and they secrete a waxy material called cerumen, which is more commonly called just earwax. This wax helps to keep out foreign objects, including small insects, and it also inhibits bacterial growth. At the very end of the external ear, and they've cut it off in this picture, is the tympanic membrane, which is what you probably previously called the eardrum. The function of the tympanic membrane is to vibrate in resonance with sound waves, which in layman's terms means that it vibrates in the same frequency that an incoming sound wave has. Next, we have the middle ear. And so the tympanic membrane is sort of right at the border. It's considered to be the border between the outer and middle ear. So it's also sometimes listed in the list of structures of the middle ear as well. So again, the tympanic membrane, or your eardrum, its function is to vibrate in resonance with the sound waves. You also have an auditory tube. And so you don't want to confuse this with the auditory canal, so it's probably better to call the auditory canal the external acoustic meatus. But you have the auditory tube that used to be called the Gustation tube, and this one connects the middle ear with the nasopharynx, which is the upper part of your pharynx in your throat, the part that sits right behind your nasal cavity. And so because we have this connection with our nasal cavity, this allows the auditory tube to equalize pressure on either side of the tympanic membrane. So you have atmospheric pressure on this side, and if you had no outlet on the other side, then you could get a very big pressure difference across the membrane. If you've ever flown in an airplane with a stuffed up nose, then you've experienced this, and that pressure that builds up, it can be very painful. So having an open auditory tube allows that pressure to equalize, and that is a feeling of like your ears popping that you sometimes experience. However, because we do have a connection to the nasal cavity, you can have bacteria go from the nasal cavity up into the middle ear and cause a middle ear infection called otitis media. And so this is more likely if you do have like a cold or a sinus infection. It's possible for that bacteria to go up backwards into your ear. In the middle ear, we also have the 3 tiniest bones in the body. These are called the auditory ossicles. There are 3 of them in each ear. So each ear has one called a malleus, which is Latin for hammer because it looks a little bit hammer-shaped, and it is actually attached to the tympanic membrane in 3 places. The malleus then attaches to one called the incus. The incus is Latin for anvil, so like the hammer hits on the anvil, and it's going to be located between the malleus and the stapes. And then the last one is called the stapes, and it looks like a stirrup, and in fact stapes is Latin for stirrup, and it's going to be attached to the incus at one side and the oval window on the other side. We also have 2 very small muscles inside the middle ear, the tensor tympani and the stapedius. These can contract to adjust the sensitivity of the tympanic membrane and the stapes, and this can allow us to be protected from very loud noises that might otherwise cause all of these structures to shake and move too rapidly back and forth. So the inner ear is composed mostly of this large structure called the labyrinth, which is a winding passageway full of canals. The labyrinth contains structures that will allow us to sense both equilibrium and hearing, and we'll talk about both of those in this part of the lecture. There is a bony portion which is continuous with the temporal bone that surrounds it. And then there is a membranous portion, and as you'll see on the next slide, so there are actually tubes inside this larger tube. The labyrinth has 3 main areas. We're going to introduce them here and then go into more detail in later sections. The first one are the semicircular canals up here at the top. You have 3 semicircular canals in each inner ear, and each one of the semicircular canals is in a different plane of space, so like the X, Y, and Z planes, or you can also think of it as like your coronal, sagittal, and transverse planes. These allow us to have our sense of rotational movement, which is part of our sense of equilibrium. The next major structure is called the vestibule, which is this swollen area right here at the base of the semicircular canals. The vestibule contains two structures called the saccule and utricle, and again we'll talk more about those later. And this is the area where you're going to sense acceleration and gravity, which are also part of your equilibrium sense. And then the final part of the labyrinth is the curly part down here that looks like a snail shell. This is called your cochlea, and this allows you to have your sense of hearing. So if we look at the inside of the labyrinth, now you can see the tube within a tube structure a little bit better. So the bony labyrinth is the outer bony part. Then you have a fluid-filled chamber, and then you have a membranous hollow chamber that is itself also filled with fluid. So perilymph is going to be the fluid that is between the bony portion and the membranous portion, and perilymph has a very similar composition to cerebrospinal fluid. Then inside the membranous portion, which would be inside all of these blue canals you see up here, would be endolymph. And endolymph has its own unique composition, so it does differ in composition to perilymph. If you notice, you've got this blue tube that runs through the semicircular canals. It's also in the vestibule where it is swollen into these different areas. This is the utricle is this swollen area, and the saccule is this swollen area. There's a swollen area at the base of each of the semicircular canals called a ampulla. And then you can notice that you have this blue canal that goes into the snail shell where it's going to curl all the way around. So once this tube goes into the snail shell or into the cochlea, we then call it the cochlear duct. There are also two windows in the bony labyrinth. So here's another picture of the bony labyrinth down here. And I like this picture too because it color codes the three main areas. So you can see the semicircular canals in blue, you can see the vestibule in green, and you can see the cochlea in yellow. And you can also see the oval window and the round window. And the oval window, remember, is where the stapes is actually going to connect. So a lot of times when you look at pictures, you can't see it because the stapes is in the way. But in this little image here, they have removed the stapes so you can see the two windows. And these two windows work together to create pressure waves in the fluid of the perilymph that goes inside the cochlea. And we'll see how that helps us with our sense of hearing a little bit later. So in this section, we're going to look at the structure of hair cells. These are the sensory receptors for both of your senses of equilibrium and hearing. So that's good news. You only have to learn the structure of one type of receptor, which can pick up all of these signals. So hair cells are specialized sensory receptors for gravity and acceleration, which remember is located inside the vestibule, for rotation, which is located inside the semicircular canals, and for sound, which is located inside the cochlea. So here is a simple drawing of a hair cell. So we have the main cell body down here with the nucleus, and then coming off of the hair cell are little extensions which give it its name, because under the microscope it looks like these cells have hair. Also notice that hair cells do not regenerate. Damage to them is permanent. So exposing yourself to a lot of loud sounds, like at loud concerts, listening to headphones with the sound very loud, can damage the hair cells in your cochlea and can lead to premature deafness. So these little extensions coming off of the one side of the hair cell, they come off on the apical or free surface, and each hair cell has between 80 and 100 of these extensions, and they're called stereocilia. And they actually resemble long microvilli, and they are shorter on one end, and they get longer as they go toward this really tall, long cilia called a kinocilium. So this is a single long cilia present on the ends of the hair cells. And then you have the stereocilia, which decrease in length as they move away from the kinocilium. So there are ion channels that are embedded in these stereocilia, and these ion channels will open and close with the movement of the cilia. So there are actually little proteins that link each of these little cilia together, and so when they wave back and forth, this will open and close ion channels, which will either allow the passage of ions or block the passage of ions, which will change the resting potential of the hair cell. Remember, the whole goal of sensory systems is to transduce a stimulus into an electrical potential. So hair cells transduce equilibrium and hearing information by moving the cilia and opening these ion channels. So the movement is going to cause a graded receptor potential within the hair cell. So here's another view of a hair cell, and again we can see the stereocilia, we can see the kinocilium, and then note that the hair cells are also attached to sensory neurons. And so as I just mentioned, distortion of the cilia can cause these ion channels to open. And if the cilia bend in the direction of the kinocilium, so in this image, if these guys all bent to the left, you would get depolarization. So the inside of the cell would become more positive. And if the cilia bend in the opposite direction of the kinocilium, so in this example, if all of these cilia bent to the right, then this would cause hyperpolarization, where the inside of the hair cell becomes more negative, and this would cause inhibition of the sensory neuron. So again, the hair cells are monitored by the dendrites of sensory neurons. So we can see, if we hook up electrodes to both the hair cell and the sensory neuron, we can see that if we bend away from the kinocilium, we get inhibition, and that blocks action potentials. And if we bend in the direction of the kinocilium, we get excitation, which causes action potentials. So now in this section, we're going to look at equilibrium, and specifically we're going to look at rotational movement, which is sensed by the semicircular canals. So if we look within the semicircular canals, they all have these membranous ducts that are made up of the membranous labyrinth, and so these are called the semicircular ducts. And as we mentioned earlier, there are 3 of them in the 3 different planes of motion, but they are named the anterior, posterior, and lateral. At the base of each of these semicircular canals, where they connect up with the utricle of the vestibule, there is a swollen region called the ampulla. And if we take a closer look at the ampulla, we can see that the hair cells are all grouped together in a region called the ampullary crest. Each ampullary crest is then bound to this light purple area here called the ampullary cupula, which is a gelatinous mass that sits right above the crest. So kind of think of the cilia that's sticking out of the hair cells as being embedded in a small mass of jello, which sticks up into the surrounding endolymph. So how this sense works is that when you rotate your head in any one of the 3 directions, this causes the endolymph inside the ducts to start moving around because of inertia, and movement of the endolymph pushes that cupula back and forth. And because the cupula has those cilia of the hair cells embedded in it, when it moves back and forth, it causes those cilia to move, which, if you remember, that causes ion channels to either open or close. So the bending of the cilia will cause either excitation or inhibition of the sensory neurons. Remember, if they bend toward the kinocilium, it would be excitation, and if they bend away from the kinocilium, it would be inhibition. So I'm going to show you a quick little video that can hopefully help you visualize what's going on a little bit better and see how this gives you your sense of rotational movement. So I have this video here, and I've turned the sound off because I just want to show you so you can visualize what's going on. So they put 3 little green balls in here so you can visualize how the fluid moves with the head movement. And so as the fluid moves around in the semicircular duct, it's bending that cupula area, which is then sending signals by bending the cilia of the hair cells. So that was in one direction of movement. Now we're going to see a different direction of movement. So like nodding your head up and down will move fluid in this way, in this particular canal. And again, when the fluid runs over this cupula area, it will cause the cupula to bend, which will stimulate the hair cells. And again, they just put the 3 little green dots in there to help you visualize the fluid movement, because otherwise it could be difficult to visualize the fluid moving. And so this is the 3rd plane and the 3rd type of movement, head movement, that it will sense. So hopefully that helps you understand it a little bit better. So in this section, we'll look at the other part of your equilibrium sense, which is your ability to determine acceleration and gravity. And the structures for this are located in the vestibule. So as we mentioned before, the 2 chambers of the vestibule are called the utricle, which is at the base of the semicircular canals, and then the saccule, which is the smaller chamber that's offset to one side. There are 2 regions, one in each one of these chambers, so one inside the utricle and one inside the saccule, and these regions are called maculae, and these are where the hair cells are located. The maculae that is located in, or the macula that is located inside the utricle is oriented horizontally, and the one that is inside the saccule is oriented vertically. If we take a closer look at each of these maculae, is shown up here at the top part, you can see that the hair cells that are located in this area have their cilia embedded in a gelatinous mass, and this is very similar to what we saw in the ampulla of the semicircular canals. This gelatinous mass here has a name called the otolithic membrane. And then a difference here is that sitting above this gelatinous mass are a bunch of tiny little calcium carbonate crystals called otoliths. And otoliths is actually Latin for ear stone. So otto is the root word for ear, and lith is the root word for stone. So these literally mean ear stones. Note that in these pictures you can also see this little duct called the endolymphatic duct that leads up to this endolymphatic sac that sits off to the side. Just as a side note, this is where the endolymph is made. So new endolymph is made here, and old endolymph is reabsorbed. So the way that the maculae work is that when you have acceleration and gravity, which we'll look at gravity first because you can test that one while you're just sitting still. If you were to bend your head backwards, for example, the gravity is going to pull on those little stones and cause them to slide backwards, which is going to cause the gelatinous material to move, which is going to bend the cilia of the hair cells, and you will get either depolarization or hyperpolarization, so depending on the direction they shift. So remember, if they shift toward the kinocilium, it's depolarization. If they shift away from the kinocilium, it's hyperpolarization. So in this example, he is looking upward, and so the hair cells are shifting toward the kinocilium. It would be the opposite if he were to tilt his head and look down. Because you can get a different signal in each direction, this lets the brain know in which direction the movement occurred in. And because you've got both the utricle and saccule with the maculae arranged in slightly different directions, all of that information together lets the brain know exactly what position your head is in relative to gravity. And then the acceleration works very similarly. So you know how if you press your gas to accelerate your car really fast, everything in your car kind of slides backwards, like it slides off your dashboard and whatnot. The same thing happens in your inner ear. The gravity or the force of the acceleration of you moving forward causes your little otoliths to slide backwards. And same thing if you slam on your brakes, which is deceleration, and everything in your car then moves forward. The same thing happens in your inner ear. The otoliths will move forward. So I have a quick little video demonstration of this one as well. So this is just a quick little clip to show you, so if you bend your head forward, the gravity is going to pull the otoliths forward, which is going to move the gelatinous mass, which is going to bend the cilia of the hair cells. And just a final little aside in this section, motion sickness occurs when the movement that is being sensed by the vestibular area and the semicircular canal area of your inner ear does not match the visual signals that come from your eyes, but we're still not sure why it affects some people much worse than others, and some people aren't affected at all. So now we're going to look at the pathway of information for our equilibrium sensory input. So we'll take a look again here at the inner ear structures. So remember that the semicircular ducts within the semicircular canals control or allow us to sense rotational movements of the head. And then we have the utricle and the saccule, which allow us to sense gravity and acceleration. So at the base of each of the semicircular canals, remember we had the swollen area called the ampulla, and this is where we had the hair cells. So notice that you have 3 of these regions, and from each of them you can see the axons of the sensory neurons coming out. And then within the utricle and the saccule, we had these 2 areas called the maculae, where the hair cells were located. And again, you can look carefully and see the axons of the sensory neurons extending outward. So the axons from all of these sensory neurons in these 5 areas come together to form the vestibular nerve. So if we follow this pathway, so again, we have these axons coming out of the 3 areas at the base of the semicircular canals and the 2 areas in the vestibule, and all of these axons come together to make up the vestibular nerve. And the vestibular nerve joins up with the cochlear nerve, which we'll talk about in the section on hearing. They join together to form the vestibulocochlear nerve, which is cranial nerve number 8. Then the information is passed to the vestibular nucleus, which is on the border between the pons and the medulla oblongata. Then from the vestibular nucleus, your equilibrium information goes to a few different places. A copy of it gets sent to the cerebellum. If you remember, we discussed in chapter 15 about how the cerebellum gets information from your visual input, from your equilibrium information, and it also gets copies of your motor commands and copies of your proprioceptive information. So this is the equilibrium information going to the cerebellum. You also have pathways that go up to the cerebral cortex. This allows you your conscious perception of your sense of equilibrium. You also send signals downward into the vestibulospinal tracts, which are motor pathways that adjust your motor tone, your muscle tone, in response to changes in balance and equilibrium. And then there is also copies that go to the superior colliculus, which help with your reflexive eye movement. So as your head is moving, your eye has to change position to focus on whatever you're looking at, and so the superior colliculus will help with that. So that is the pathway of equilibrium information. And at last we come to our last special sense, our sense of hearing, which takes place in the cochlea. So a quick review of sound so you can understand how the process of hearing works. Sounds are actually pressure waves that travel through the air. So like if you have a sound coming out of a drum, for example, the sound itself is actually compressing air molecules in some areas, which then decompress or cause them to be less dense in other areas. So it's actually variations in the air pressure is what makes up a sound. And here is another image to your right. It shows the motion of the air molecules. So when air molecules are pushed closer together and then farther apart, it creates these waves, and those are sound waves. So we can measure sound waves in frequency and amplitude. So the frequency is how frequently we get these waves per a given point of time. And so we usually measure frequency in hertz, which is the number of waves that pass a particular point per second. So how many waves pass this point per second would be the frequency. And humans perceive frequency as pitch. So when you talk about high-pitched sounds or low-pitched sounds, you're talking about different frequencies. Humans can hear between 20 and 20,000 hertz. Those are the frequencies that we are able to detect, at least when we're children. As we get older, the range of frequencies that we can detect narrows as we have cells within our cochlea start dying. Amplitude, or the intensity of a sound wave, is measured in decibels, right? And so that is like how dense the air molecules are in one area compared to the other. So the bigger the difference, the larger the amplitude. And loud, very loud sounds would have a very high decibel level. All right, so let's look at the anatomy of the cochlea a little more carefully. So as we mentioned before, the cochlear duct is going to be the part of the cochlea that is filled with endolymph, and it's going to swirl around the snail shell. And then in this top picture here, it's shown as the blue line that is swirling around the cochlea. And it is actually sandwiched between two other chambers. So because, remember, we have the bony part that's on the outside, and then the cochlea is on the inside of the membranous part, we still have fluid that is between the bony wall and that cochlear duct. So this fluid is divided into two separate compartments or chambers. We call one of these the scala vestibuli, and again, this is filled with perilymph. And the scalar vestibuli starts at the oval window, and so in this picture up here, it would be the purple arrows. So it's going to travel around, right? And when we look at a cross section like this, the cochlear duct are these little blue areas that you see on the ends, and the scala vestibuli would be the circle above the cochlear duct. This can also be called the vestibular duct. And then when you get to the very tip of the cochlea, so the tip of the snail shell, and you go down the other side, and that's shown by the red arrows in this top picture, that is called the scala tympani. So it travels from the tip of the cochlea down to the round window, and when we look at this cross section down here, the scala tympani would be these chambers below the cochlear duct. And these are sometimes also called the tympanic ducts. It can be a little hard to visualize with the way the textbook has it drawn, so I have another picture here for you to look at. So again, I have the same information here, but if you look at this top image up here, the snail shell has been unwound, which makes it a little easier to see. So in this top image, the cochlear duct, which is also called the scala media, would be this middle skinny purple line. And then sounds come in through the oval window, which is under the stapes, and they travel upward. That would be the scala vestibuli. And then at the very top, they cross over and travel back down, and that would be the scala tympani, and ends at the round window. So if we look at a cross section of this, the cochlear duct is this little area over here that's purple, right? It matches that little purple line up here. And then underneath it would be the scala tympani. Above it would be the scala vestibuli, and sounds come in the oval window, travel up the scala vestibuli, reach the end, wrap around, and travel back out through the scala tympani. And then here's the cross section from your textbook. So when you're looking at the cross section, we see the cochlear duct here in the middle. That's why it's called the scala media. Media means middle. Above it would be the scala vestibuli, and below it would be the scala tympani. And then we're going to talk about this special area here in just a second, because this is where the hair cells are located. So in conclusion, fluid waves are entering at the oval window. They travel up the scala vestibuli to the tip of the cochlea, and then they travel down the other side, down the scala tympani, and they exit at the round window. So a lot of students have a little trouble actually visualizing the oval and round windows and what they're doing. So the reason that you need both of these windows is because liquids don't compress. When you push on a liquid at one end, the other end has to go somewhere. So think about squeezing a water balloon. If you squeeze one end, the other end's going to bulge outward. And so you can see this too if you play around on a water bed, right? If someone goes in one side, it's going to push another part of the water bed out on another side, right? And so that is the purpose of having these two windows. They work together to allow the creation of fluid waves in the perilymph of the cochlea. So basically, when the stapes pushes in on the oval window, that causes the round window to bulge outward. And then when the round window bounces back and goes in, that causes the oval window to bulge outward. So it works just like a water bed or a water balloon that's being squeezed. All right, so let's look at the spiral organ. So this is the location of the hair cells. This is called the spiral organ. It also used to be called the organ of Corti. And so we have a close-up view down here. And so let's see where we are actually looking. So this is located inside the cochlear duct. So it's sandwiched between the scala vestibuli and the scala tympani. So hair cells sit on this part called the basilar membrane. So the basilar membrane is the membrane at the bottom. So think basal membrane. The hair cells are embedded into this membrane. And then the cilia of the hair cells are connected to another membrane called the tectorial membrane, which stretches out over top. So again, you can see it up here in this image. The hair cells are embedded in this blue part, and the tectorial membrane is this red part sticking out here. So as we'll see in a moment, sound waves make the basilar membrane bounce up and down. And so if you can imagine, when it bounces up and down, it's going to push the hair cells into the tectorial membrane. The tectorial membrane does not move, so when the basilar membrane bounces up and down, it's going to push those hair cells and it's going to bend the cilia as they crash up against the tectorial membrane. And remember from before, anytime you bend cilia on hair cells, it causes the opening of ion channels, which causes a graded receptor potential, which can lead to an action potential. So it's the same mechanism that we saw with the equilibrium, only this time the cause of the hair cells bending is the movement of the basilar membrane pushing those cilia up against the tectorial membrane. So we've arrived at the 6 steps involved in being able to hear sound. So step 1, the sound waves come in through your external acoustic meatus and they arrive at the tympanic membrane and they cause the tympanic membrane to vibrate in the same frequency as the sound waves. So we say the tympanic membrane matches the resonance of the sound waves. Step 2, the movement of the tympanic membrane is going to move the 3 auditory ossicles. So the tympanic membrane is directly connected to the malleus, which is going to move the incus, which is going to move the stapes. Step 3, movement of the stapes up against the oval window is going to produce these fluid pressure waves in the perilymph of the scala vestibuli. And so again, remember, it can do that because it's also going to bulge out at the round window. So the stapes is pushing in at the oval window and starting these pressure waves. Step 4, the pressure waves are going to cross over the basilar membrane, and the specific area where they'll cross we'll talk about in a moment, but at some point along the trip up the cochlea, the pressure waves are going to distort the basilar membrane, and that's going to cause the basilar membrane to bounce up and down, right? And then, of course, the pressure waves are then going to exit through the scala tympani and out the round window, but we're focusing on the basilar membrane. So step 5, the vibration or the movement of the basilar membrane up and down is going to cause those hair cells attached to the basilar membrane to be pushed up against the tectorial membrane, which is going to cause the cilia to bend, ion channels to open, and an electrical signal to be generated. And then step 6, the sensory neurons are going to pick up that electrical signal, and they're going to carry it to the central nervous system through the cochlear nerve. So that is how we hear sound. So how can we differentiate between the frequency and the intensity of sound? The way that we can hear a frequency, so whether it's a high-pitched or low-pitched frequency, has to do with the part of the basilar membrane that bounces up and down in response to the fluid waves. So high-frequency fluid waves cross over very close to where they enter. So they enter at the oval window and they immediately cross over and go out the round window. So very high frequency sounds activate the basilar membrane at the base of the cochlea. Low frequency sounds are going to travel farther up the scala vestibuli before they cross over. So low frequency sounds are going to activate the basilar membrane towards the tip of the cochlea before they cross over to the scala tympani and then go out the round window. So the specific point on the basilar membrane that responds to a specific frequency is how our brain knows the difference. So a signal coming from this side will be interpreted as a high frequency sound, and a signal coming from the end of the cochlea would be interpreted as a low frequency sound. The intensity of a sound is coded by the activity and the number of hair cells activated. So a really loud sound is going to make that basilar membrane bounce more forcefully and push the hair cells up against the tectorial membrane more forcefully. That'll cause more ion channels to open, and it'll give you a larger depolarization and a greater signal. You'll also get more hair cells that will be pushed up against the membrane. So not only will the ones that are being pushed be pushed harder, but you'll have more hair cells also being pushed up against the membrane. That's how the brain knows that it's a loud sound versus like a whisper. Notice though that loud sounds can damage hair cells because it is pushing them up against the tectorial membrane so forcefully. This is why you don't want to be exposed to very loud sounds for long periods of time, because it will permanently damage your hair cells. And damage to the high frequency hair cells, so the ones closest to the base of the cochlea, happens first. Young children can hear the highest frequencies, about 20,000 hertz, but as you get older, you lose your ability to hear very high frequency sounds. They actually took advantage of this a decade or so ago. People created ringtones for cell phones that only young people could hear and older people couldn't, so they made it a very high frequency ring. And that is the end of how we hear. So we just have one last thing to cover. And the final section of this chapter will be about the auditory information pathway. So we start with the hair cells in the cochlea, and they're going to send their information to sensory neurons, and the axons of these neurons come together to form the cochlear nerve. The cochlear nerve will join up with the vestibular nerve to become the vestibulocochlear nerve, which is cranial nerve number 8. The part of the vestibulocochlear nerve that is carrying sound information is going to go to the cochlear nucleus within the medulla oblongata. And then from the cochlear nucleus, it's going to go upwards to an area called the superior olivary nucleus. Then it's going to travel up to the inferior colliculus, where we're going to have our auditory reflexive actions take place. So remember, the inferior colliculus controls auditory reflexes. From there, it's going to pass up to the medial geniculate nucleus, because all sensory information except for olfaction passes through the thalamus. Keep in mind that vision goes to the lateral geniculate nucleus and auditory information goes to the medial geniculate nucleus. From there, it's going to go up to the primary auditory cortex in the temporal lobe. And we do have a map in this cortical region as well. So the different areas of the basilar membrane and the cochlea are mapped out to different areas within the primary auditory cortex. And that is the end of chapter 17. So aren't you glad we only have 5 special senses?